Hi, today we're going to learn all about the microscope so that you are ready for lab next week. So this is a microscope. This is the base of the microscope right down here. This is the arm of the microscope, and those are the two parts that you would carry it by safely. So you would pick up one hand on the arm and one hand on the base to ensure that you're supporting it and not likely to drop it. From the arm, we have the head. The head is where you're going to find the ocular lenses. The ocular lenses are where your eyes go. There are two ocular lenses because we have two eyes. So these ocular lenses are adjustable in order to fit the difference between your two eyes. Also, the left eye piece has a focus on it to help you focus in for your particular eyesight. So in order to use those ones, you would look at your specimen with your right eye closed and make a very sharp image with your left eye before you open both. After your ocular lenses, you're looking through the head down into the nose piece where your objective lenses are located. There are 4 objective lenses. There is the scanning power, which is also 4x magnification. There is your low power, which is 10x magnification, your high power, which is 40x magnification, and your oil immersion lens, which is 100x magnification. In addition to these magnifications, your ocular lenses have 10x magnification. So really, when we're looking with the scanning power, which is 4x, we're multiplying that by the 10 here. So we're seeing the item at 40, the specimen, at 40 times magnification. Okay, and again with your low power, which is 10 times 10 is 100. So we're looking at the specimen at 100 times magnification. After your objective lenses, we have the stage right here. The stage is where you place a microscope slide. On top of the stage are clips. I'm going to hold it slightly angled so you can see. So there is a clip right here with a little button to open it up. When you place a microscope slide on your stage, you want to make sure you're in the clip. So open it up, place it nice and flush on the stage, and close it, and it holds it in nicely for you. You can also move the stage clips with these 2 buttons right here, these 2 knobs. One moves it left and right, the clips left and right, and one will move the stage back and forth. This will help you center the specimen on your stage. From there, we have the light source all the way at the bottom on the base and the light source control right here. And this will change the intensity of the light power. The on button is on the back. So I can flick that on. The light source illuminates, and I can change the intensity. So this is the lowest setting, and that's the highest setting. There are other ways to adjust the light. There's something called a condenser, which is this unit right here, and I can raise and lower the condenser with this knob. And in general, the best setting is to be as close to the stage as possible or all the way up. And again, that's just going to control or focus the light on the specimen itself. On the condenser, we have the Irish diaphragm, and you can see that light changing in intensity up here as well. This is going to open and close to change how much light is actually going to hit the specimen, and this gives us good contrast. So the best position to start off is half open, half closed, and then you can adjust it accordingly as you see fit. But you want to always try to start with the iris in the halfway mark and the condenser all the way up. I also usually keep my light source on the highest setting as well to start. Those are all the pieces of the microscope. Then we have the adjustment knobs. The coarse and the fine adjustment knobs are located in the same region or the same spot for this particular microscope. The coarse adjustment knob is on the inside and the fine adjustment knob is on the outside. Both of these knobs move the stage up and down this way, closer to the objective lenses or further away. The coarse adjustment moves it quickly, and the fine adjustment only moves it in very small increments that you can barely see with the naked eye. You only ever use the coarse adjustment knob when you are in the scanning power. You want to get that as close to focus, the specimen as close to in focus as possible. And then, once you want to use the fine adjustment to really make sure you take away any of those blurry edges. You want to have the crispest image possible before you move on to low power. Once you're in low power, no longer use the coarse adjustment knob, only use the fine adjustment knob. This will make sure that you're not losing your image completely. So to start, we want to start with our microscope in scanning power, and we want to place our slide, which I already did, on your stage, in the stage clips so it lays flush. You want to try to make sure that the specimen is centered. The specimen that I'm using is a pre-prepared slide with only one small dot in the center. So I want to make sure that that is in the center so I can see it right away. So right now all I see is a bunch of pink. My condenser's all the way up. My iris diaphragm is halfway open and my light source is all the way on. I move to the stage all the way up with my coarse adjustment knob, and I'm going to adjust slowly from there. So this particular sample is already pretty close to being focused in. By the way, I'm using both eyes to look. I'm making sure that the spacing between the ocular lenses is adjusted so I see only one circle, because I need my binocular vision to bring those into one view, to mesh the images into one circular view. I just adjusted the coarse adjustment a tiny bit, a slight adjustment, and I see the image pretty crisply. I'm going to adjust my fine adjustment just slightly to take away any blurry edges I might see. At this point, I feel like I'm as focused as I can be, and I'm going to move to low power. In low power, remember, I'm only going to touch the fine adjustment knob. Very small amount of movement was necessary in order to get a crisp image. Now that I'm zoomed in on low power and I'm still centered, my image is still centered, I'm going to move to high power again. If your image is not centered, once you zoom in, if it's maybe off to the side a little bit, make sure you adjust it, because you could potentially lose your image completely. I'm going to move to high power, and you can notice that the lens is very close to the microscope slide. This microscope slide is pre-prepared, so it has a cover slip on it, and I'm not going to use oil immersion. And we'll talk a little bit more about that in 1 second. I just moved to high power. The image is still there, but it's not clear at all, so I'm going to adjust my fine adjustment again. And now I've been able to pull in a very clear image. Right now I'm looking at frog skeletal muscle, so I can see the muscles, I can see the nuclei in each of those muscle fibers. And if I needed to take a picture, I could take my cell phone and put it up against one of the ocular lenses. And if you hold your arms, especially with your elbows on the table, to steady them, you should be able to get a pretty good picture of what you're viewing. And that's going to be important for our lab, and where I'll have you take pictures of what you see and put them into a Google presentation so that I can make sure that you're actually seeing the specimen and aren't just thinking you saw them. A lot of times what happens is some of the early steps of adjusting, you can end up actually trying to focus in on the layers of the glass itself of your microscope lens, of your microscope slide rather. And so you're looking at kind of the crystals that form the glass, and that gets very confusing. So if you're having trouble, please call me over. But the reason why I want the picture is so that I know that you're actually looking at the right structures and not structures of glass. Once you're in high power, you can't go any further because oil immersion requires oil in order to put a drop of oil on the microscope slide. So you would move the lens to halfway, you would put a drop of oil, and then you'd slide in your oil immersion lens. The oil is going to help magnify, but it also helps protect the lens and the slide from scratches. So never use the oil immersion lens unless you are truly doing an oil immersion on your slide. Do not even move past the oil immersion to get back to your scanning power. You have to go in reverse, because this, as you pass it through, could end up scratching the microscope slide, or more importantly, it could scratch the microscope lens. So we always want to go in reverse, or you can take that slide out, you can lower the stage, and then you could go past. That is how we use a microscope, how we focus in. The last thing that we want to do is make sure we clean our microscopes, and we use a special a special lens paper for it, which I took out and then I forgot to have close enough, so I'm going to grab it. So the special lens paper right here, it says lens paper right on it. You can tear one of these out. We also have blue boxes that's more like a tissue box where you pull from the top, and you're going to use this to wipe down your eyepieces. And if you had done oil immersion, you would use this to wipe the oil immersion lens down really well. We can also use isopropyl alcohol or rubbing alcohol to spray this and clean your eyepieces in between use. When you're done, you want to make sure that you're back in scanning power, that you shut off your light source, you unplug, and you wrap your cord around the base here. And then again, you put a cover on it, and we carry it by the arm and the base. So that is the general use of a microscope, and hopefully that will help us get started right away in our lab next week. Thank you.